Okay, so I hope you can all hear me all right. Thank you for inviting me. It's a real pleasure to be here to talk to this audience. So some of it's quite basic. I was told sort of first year undergraduate level biology understanding, and then some of it's more complicated towards the end. So I hope I don't lose anybody on the way, but I can always re-explain or go more slowly, etc. So just let me know if you get stuck anywhere. So I thought would be quite important is just to, because I run the toxicology unit, it's not all about poisons. This is to explain what we do and why the work we do is really important. So, this is a sort of definition of it from the UK Society of Toxicology. So it's a study of the adverse impact of chemicals, physical, biological agents on living organisms, but actually understand prevention and amelioration of such effects. So you understand what the toxic problem is, but how you might overcome it. And that's particularly important within drug discovery because it's an iterative process. So you need to have the tox up front with the development, or else you end up with what says a valley of death in the drug design, that you've made something you think is wonderful, then you find right at the end process it's toxic. So that's the wrong way around. It has to be done in parallel, and you can only really do that through mechanism. So having said that, you can see that actually toxicology allows people to make informed and balanced decisions, and it impacts on lots of things. So obviously on disease and disease outcomes from exposure, as I said, drug design and drug safety, and environmental exposure and risk assessment. I'm not going to show much about that, but I'm going to just use a couple of examples of how that works in terms of exposure. What's really, really important in a tox term is understanding causal versus correlation. So often you'll see things about, microplastics or bisphenol A, etc., and all these claims that it does X, Y, and Z. But actually without the mechanistic link between the two, it's not possible to make those claims or understand what's going on in either direction. So you really do need detailed mechanistic science and detailed molecular biology, which is what we do, to really drill down to get to the issues involved and how you might overcome them. A lot of young people here in the audience, one of the things that is really, really important for the UK as a whole is to train more toxicologists. So there's an enormous skills gap in this area. We have various PhD places going. If you're interested, have a look at our website. It can have a very, very rewarding career ahead, from a basic scientist working in regulation and regulatory affairs. And there's a big gap. So actually this is an amazing career opportunity for anybody. All our students get snapped up in terms of jobs, whether it's industry, regulation, etc. Some people get offered their jobs partway through their PhDs because there's not enough suitably trained people around, and particularly in the newer therapeutic work that I'm going to talk about. And these are we've got 46 students in the unit at the moment, and these are some of our wonderful students who are around. Okay, so I'm going to start at the beginning and talk about 4 basic concepts of tox, and then show how those basic concepts also apply to new therapeutics and why mechanism is important. So this is Paracelsus, who's often called the father of toxicology. He was a medic and a scientist who did some quite extraordinary work in the kind of 16th century. His real name is Philippus Aurelius Theodorus Bombaston von Hoffenheim, but his friends called him Paracelsus above Celsus, so kind of the highest great one in recognition of the work. So he was the first person really to actually treat people with minerals. So he used sort of tincture of zinc, for example, to actually work out, sometimes it made people better, sometimes they're worse, but one of his observations was it's actually very dose dependent. And so he is quoted having this phrase, which is here, dosis solo factit venenum, the dose alone makes the poison. So things that can be a therapeutic at some dose, if you go too high, they become severely toxic. All sorts of things. If you just think of paracetamol and paracetamol poisoning, one thing gets rid of the headache, another thing can have a very deleterious effect on liver and liver function. So this is an example. So this is arsenic as an example. So LD 50 is a lethal dose 50. So this is how much you require to kind of kill somebody. Is on an average be 30 milligrams per kilogram. And it actually became a favoured poison in the Middle Ages because it's very hard to detect. And then when you have the symptoms, they're actually quite similar to those of cholera. So they could claim somebody died of cholera not being poisoned. And actually, in that period, it had the nickname inheritance powder. so you want to bump off your elder relatives, this was the way to go. And this is a slightly grim picture, but these can also, you can get sort of lesions on hands, can you get similar things with acute cholera as well. But on the other hand, this is at high doses, but if you take the dose down, you can therapy. And traditionally in Chinese medicine, it was used for 2,500 years, it's used therapeutically. And actually in the Western world, it was used to treat syphilis before antibiotics were introduced. And I mean, very toxic to have as a treatment, but it did give cures from syphilis in that time that would give a death sentence otherwise. So it's dose, that's the very key thing. The next thing I think is important as well is actually understanding hazard versus risk. And I'm sure you're all scientists, so you're aware of this concept, but often the general public often don't grasp this, and it's very easy to see these extraordinary headlines in the tabloid newspaper about the hazards of things that are not really a hazard or a risk. So the hazard is potential for harm for something, and that's the intrinsic property of the chemistry of whatever it is. And the risk is really the chance that this is going to actually happen to the person. So the hazard, you have the risk really is a hazard times the exposure and the dose. Oops. So the degree of hazard is really, say it again, a combination of how common a substance is and how likely you are to be exposed to it. So just to have, not jokey because it was horrible for the people, but some examples. So you will have heard probably, for example, of Novichok. So these were a group of nerve agents made in Russia in kind of between 71 and 93. That's part of the structure of it. So it's an acetylcholinesterase inhibitor, has incredibly toxic at tiny doses, and then it leads to involuntary contraction of skeletal muscle, respiratory and cardiac arrest. And these are the Skripals, and this was painted onto their handle of their house, and then they recovered but was very badly damaged by it. And then there's a very poor woman who picked up the spray that had it in and then sprayed herself, and she died as a consequence. So there was a terrible thing happened in Salisbury. Another one, it's all spiralated. This is Alexander Litvinenko, who was given tea by a Russian agent in a hotel that was laced with polonium. So this extraordinary toxic by mass is 250,000 times more toxic than cyanide. So you don't need much polonium to kill you. And of course, once it was in, he died from radiation poisoning. And the next one, which you will be far too young to remember, but I remember this very clearly, this is Grigory Markov. And he was a spy living in London, and he was injected with a pellet containing ricin from an umbrella. He was stabbed with an umbrella. And then the pellet it was pretty grim. It had, they worked it out, it actually had like a wax coating on the outside, and as the wax melted, then the ricin was released into his system. It's all very James Bond stuff. So these things, look, 1,500 times more, these things are incredibly toxic. But again, the chance of being exposed to Novichok polonium is incredibly small. And so the chronic exposure, though, to metals and other agents are more of a serious issue for many people around the world. And if we go back to arsenic again, particularly, for example, in some low middle income countries and in parts of India, pollution of arsenic in water is a very serious issue. And this is just showing the arsenic, the percentile of arsenic and chance of being exposed to this within the UK. Okay. So arsenic, as I said, has got an LD50 of this. It's a fraction of those really, really terrible things. But actually, on the toxic substances and disease registry, it's at the top, because you're much more likely to be exposed to elemental arsenic than you are of those things are much, much lower down the list. So it's a question of understanding the mixture between the hazard and the risk, and that's an important concept in environmental tox. But I'm not going to talk about any more environmental stuff. That was just to get the process across. So the other concept, of course, and you'll be aware of this as scientists, is the risk-benefit ratio, and that's really, really important concept in medicine. So all medical interventions are associated with risk. Taking paracetamol is a risk, taking ibuprofen is a risk. as individuals, every time you take a drug, you're doing an experiment on yourself, because no two people respond to any one drug in exactly the same way. As an average, a population may mostly respond in the same way, so you know that things are safe, but it will not be identical because we're all different. So alternatives exist for all intervention, including no intervention, and these likewise present their own risk-benefit. So was it a benefit to do something versus not to do something? Is a risk-benefit? So clinical decisions, you compare and contrast that risk ratio of benefits to the alternative thing, and that gives you the risk-benefit ratio. And so some risk-benefit ratios are going to be very disease-dependent. So, for example, individuals who have cancer, a lot of the cancer treatments, as you'll know, are incredibly toxic chemicals you give. And so whilst people have awful side effects from them, and that can be clinically managed, but even so it's very debilitating, in a lot of cases that you put up with the side effects because the benefit is the eradication of the cancer. So that's your risk-benefit. But for a normal drug, you'd never accept to have those sort of side effects for a normal drug. And that's a very important concept within the tox field, is a risk-benefit ratio. When you're judging a medicine, what is the risk for harm versus the benefit to the patient? Okay. So this is the other one, and this is the big problem. This is off-target drug reactions. So every drug has two reactions. There's nothing totally clean. One you know about and one you don't. And the one you know about, it will just be an exaggerated pharmacology, so you can judge against that and work on dose. The one you don't know about is a problem. And so it's not predictable from knowledge of the pharmacology. There's very, very difference between individuals. The other word is used is idiosyncratic toxicity, that's individual dependent tox, and there's a very complex dose relationship. So actually, adverse drug reaction is the chemistry of the drug and the biology of the individual. And we can test for that, but it's very hard to test for this. This is another big issue within the tox area. And if you don't have enough very, very detailed mechanistic information, precisely what pathways are initiated with the drug in response to exposure, you can't really start to answer that. And that's kind of where the work that we do in the unit comes in, because it's getting down to the mechanism of drug action on cell and machinery, so you have a better chance of predicting off-target tox, even within a very diverse population. And so, if you look at this from a drug point of view, so to get a drug even to trial can cost billions of dollars. It's a very, very expensive process. And as you can see, you can have these various causes of attrition, and these are adverse drug reactions. And the most common organ for common drugs is actually cardiovascular tox and hepatotoxicity. So often you see liver toxicity because that's where you have everything ends up in the liver. That's where you have the greatest accumulation. So these things are bad up to here. This is a disaster if you have post-marketing withdrawal. And so these would have been tested. They would have gone through all the clinical trials and they look fine. But that's when it comes into, when you roll it out into a bigger population, that's when the biology of individual comes into effect. So they might be fine for some, but not for others. So what about other things going forward? But then we've only really scratched the surface in developing drug treatments. We've got 7,000 rare diseases, and 95% of those don't have any treatment options at all. We've got small molecule targeting proteins. And so everything at the moment is getting for small molecules, but actually there's other ways of doing this. And there's an urgent need for new therapeutics to come through. So that's when we come into these. So they're called various things. The Medical Research Council that I work for, my unit's funded by, calls these things advanced therapeutics. So these are not all of them, but these are the nucleic acid based ones. So we're particularly interested in nucleic acid based therapies. So they've got DNA or RNA as part of the therapeutic agent. And these are just the list of some of them. I'm going to talk about the first three, but not this one, but we can discuss that after if people want to. So there's modified in vitro transcribed RNAs. So this is a therapy that was used in the COVID vaccines. There's antisense oligonucleotides. I'll explain this in a bit. These are used, and these ones silencing RNAs. So these two are used now to treat rare diseases with some extraordinary successes but failures. CRISPR-Cas9 editing, you may have seen some of this on the news. This is actually recoding, editing the genome to actually have a design, remove a bad gene or put a good one back in again. So this is complicated to get this to work. But they're all, basically, it's using nucleic acids to reprogram the genome in some way or another. They've got the most, they're incredibly exciting. It's the potential to transform healthcare if we get this right. Currently, they're only used for rare diseases that have limited treatment options and not really been rolled out for more common diseases. So there's opportunity to do this in the future, providing we can get the toxicology and the safety science and the safety liabilities corrected. So this is very basic for some of you and not so much for others. So the central dogma of cell biology is that DNA, which codes for who we are, is transcribed into this intermediary substrate called RNA, and the RNA is translated, it's read by a machinery in the cell called ribosomes. I'll show that again later, and that makes the proteins. So for the most part, in our bodies, the proteins are the business ends. They're the things that do the various functions that the body requires, controlling everything. So if you can turn off, for example, expression of, oops, I've gone the wrong way. You can turn off expression of a protein by modifying the RNA expression. And that's what two of these therapies do. So this is silencing RNAs. I mean, if you're not a biologist, don't worry about the details. But basically, you have this double-stranded thing comes in here. It binds this AGO complex, part of the RISC complex. You get degradation of one strand. This strand is brought through by these RNAs, and they bind to your target RNA in the 3' UTR, generally, but not always. And then you get RNA degradation. And 5 drugs that work in this way have been approved by the FDA and the Medicine Health Regulatory Authority within the UK as well. And this is the other one. This is antisense oligonucleotides. So they bind to their target RNA, and this is in the nucleus. This protein here, called RNase H, comes along and it cleaves these hybrids into lots of bits. And that's 10 approvals. These can also be used for splice switching, but that's quite complex to explain in detail in the time. But again, once we can talk about that and how that works as well. Okay. So these are the successes. So this is an ASO. This is nunacerin. It's been approved to treat spinal muscular atrophy, SMA. This is a child who's been treated with this drug. So without treatment, oops, I'll just put these down. This is, it's the most common genetic cause of infant death, and it's a wasting disorder, and it leads to gradual, desperately sad for patients and for parents because the child appears fine, and then you have this gradual wasting and loss of function, and then death at a pretty young age. And this drug works by the mechanism I showed you. It halted disease progression, and 60%, not only did it halt it, but 60% of the infants treated had improved motor function. So this is a really, really big success. This is another one. This is an siRNA, so this is knocking out expression. And this is kind of like a sciencey graph thing, but don't worry too much about this. So some individuals have very, very, very high levels of cholesterol, so much so that they have children who are like 10 years old having heart attacks because of the cholesterol buildup. So it's quite rare, and there's sort of milder forms, but also comes with significant risk, and it's not possible to control these high levels with standard drug treatments. And what you can do is this. So the protein is PCSK9. You can reduce this, and that reduces cholesterol. So you target that protein by this treatment. So what this is showing you here is that what's extraordinary about these treatments, they're very, very, very long lasting. So you give them, and they last, it's not working terribly, never mind, they last ages. You can see on that graph, you give it, and they can see the bottom line underneath, it stays down for such a very, very long time, and that's matched by a change in the cholesterol level. So again, this is an extraordinary success and a massive treatment of an improved drug. The doses are quite high, and they give 284 milligrams for one dose, followed by one at 3 months and then one at 6 months. But this is, incredible. It works brilliantly for treating this. They're a bit worried at some point because actually it's so successful, they've reduced the level of cholesterol too much. So it has to be kind of slightly modified in how much they give and how much they take the level of this protein down. But again, it's another new therapy that revolutionises the lives of individuals with this inherited disease. So, why aren't they used to treat common diseases if they're so good? You could, in theory, knock up, most drugs are targeting proteins. You could change protein expression by a lot of these programmable nucleic acid therapies. Well, even on this, so this is for SMA. Have a look at the side effects of these. They're very severe. There's a lot of problems with kidney, bleeding complications, etc. So, it's the same concept that we came back to, is the risk-benefit ratio. So you might take this level of, not harm, but level of problems with it, and liabilities, of safety liabilities for something that's a very life-limiting disease. There's not really another option. You wouldn't want to have this in replace of treatment if there's other drugs with a better safety profile. So that's one reason. Okay. And then the other one is there have been a lot of failures. So we don't have this right yet, and quite high-profile failures. So very sadly, there was an ASO to do a knockdown of Huntington protein to treat Huntington's disease, and there was very bad brain tox with this drug, and that failed quite catastrophically. There was another one to treat ALS, and that failed, etc., etc. So there have been big successes, but also notable failures, and every failure is costing millions and millions of pounds because of the money that went to get it developed so far. And that's because we don't understand enough how these things are working. We don't understand the safety liabilities fully. We've got to get more mechanistic information out there. So this is just a graph to show some of the toxicities associated with these new therapies. And if you can look on here, it's the same as standard drugs, except we're having renal tox. So a lot of these things are working wonderfully, but if you look, some of them, we've got cardiac failure, hepatic tox, renal tox, a lot of failure in phase 2, phase 3 trial. We need to get better at this. There must be far better ways of predicting the tox before it even goes into patients. So you need better clinical models, better preclinical models to stop some of this from happening. In the last one on the list, that's a CRISPR-Cas9 lipid nanoparticle. The patients that survived was a very small number of patients. They were elderly. 2 people died on that trial. So it's not acceptable that we're having this level of risk from these newer treatments. We've got to have a better understanding of what's going on. Okay, so what about NATs? So we've got 2 of our standard concepts that I've shown before. So one is dose is still the same. The fact that we're getting a lot of the tox in the liver and the kidney is a dose effect. So when you take one of the reasons they're targeting rare liver diseases using nucleic acid therapies is that they all end up in the liver, all of them. So you inject them IV, they go straight into the liver. It's really a liver dose. So it's a great way to treat a liver disease. It's much harder to treat other disease because it's going to the liver. And then once they're in the liver, the way they get excreted is through the kidney, so you end up with kidney tox. So actually it's accumulated dose in those organs. It's an on-target dose effect that's causing quite a lot of that tox. So you've got to understand this better. And then there's off-target drug reactions, so they're binding to things you don't think they'll bind to. This is what I'll talk about in a bit more detail. There's a lot of action that we don't know about, so we need much better, improved ways of delivering these to the target organs, so you don't go to non-target organs, but also better mechanistic understanding about the drugs themselves. Okay. So, what sort of things happen when you give a cell a nucleic acid therapy? The cell has a big response there when it thinks it's being attacked by a foreign piece of DNA or RNA, and that's effectively what we're doing when we're giving a nucleic acid therapy. So you've got a lot of endogenous RNA sensing, so it's the same as it thinks it's being attacked by a virus. This is part of the antiviral response, and that's what you trigger in with nucleic acid therapies. One of them is part of this response here, and you activate that protein. The other one is actually this response. It's the innate immune response. So again, it's a cell. It's an immune system within your cell for sensing foreign attack. So this is either double-stranded RNA or single-stranded RNA activate these binds these proteins in the cell, and they activate a whole inflammatory process, and you end up producing interferon, for example, inflammatory things. But it can also lead to cell death. So often when cells are attacked by viruses, they'll trigger cell death as a way of defending the whole organism against viral attack. And you don't want that to happen when you start treating with a therapy. That's not what you want. It's the last thing you want. So to overcome this, what's been happening in the field, so this has been kind of transformational, is that they've started modifying the RNA and the DNA that's part of the treatment. You decorate them with different sorts of chemistry, and that changes the safety profile. So you can stop activating the innate immune response. So you go from a traditional RNA to first, second, third, and then these locked nucleic acids are a third generated antisense oligonucleotide with different chemistries on there. And that has worked really well. So it's really to damp down the immune response because they look more similar to kind of cell RNAs without modifying them. So the cell doesn't think it's being under attack. But on the other hand, it's increased the tox in the liver. So we stop one problem, and then we've got a bit of another one. Okay. And then there's a lot of sequence-dependent toxicity that we're getting to the bottom of as well. So we've got chemistry-dependent tox. There's also sequence-dependent tox. So this was a very nice piece of work that was done. And what they did was they took one antisense oligo and they changed the sequence by one base at a time. And single base changes will make one toxic versus non-toxic. But if you've got to scan each one by this much, that's going to be a nightmare to make these drugs. You start off with thousands to get down the one you want that has the function you want but isn't toxic at the same time. So we've been doing some work in this space. And this is Alex in my lab who's been doing this work. So what he did was he messed around with various toxic ones and did a whole load of things about making toxic ones safe and safe ones toxic by changing single nucleotides. And actually, won't explain about the assay because we've got a pattern thing going on, but we've got a way of screening for this in a very rapid format, but it's structure. So actually, but it's not whole structure, it's small bits of substructure are causing toxicity here. And that's why a single change, you can go from having 3 bonded pairs to 2, and that's enough to go to safe to toxic. And that's what's been shown on this slide here. And the reason for this is that we actually activate those proteins that are part of the innate immune response. So antisense oligonucleotides that are meant to be single-stranded activate the double-stranded RNA sensors in the cell because of the substructures that they're forming, and that you don't need this big blob on the end, that very highly stained bit on the top, very top one on the end. That's when you put a very, very long stretch of double-stranded RNA in a cell. That mimics a very strong viral infection. But actually, small bits are enough to do this as well. Small bits of substructure will give you not that level of activation, that's completely over the top, but that will give you small bits. If you see the darker band, it shows we've got activation of that. And then on the right-hand side, when you've got AGG, you take out 1G and you reduce that tox. So actually, we've got a way now of screening for structure-dependent tox that you don't have to make a whole thing. You can just do one could just do a rapid screen on a 3-4-well method to get rid of that. The other thing is azo-protein interactions. So initially this wasn't thought to be such a big thing because these are, we've got we've got positive and negative charges on these. And the trouble is, the charges bind proteins. And so a lot of the modification, though we're taking out some of the problems, we're getting more protein-protein interactions as well. And that impacts a lot of the performance of them. So so proteins all over your cell, and they'll take things to different places, like postcodes. If you have a protein that is in the ER, it'll take your ASO into the ER, even if you don't want it there. That's the problem. And then you might start disrupting some of those normal functions in other bits of the cell. You just take it around to where you don't want it, and that's an issue. And this is thought, these protein interactions are thought to be a key driver of some of the chemistry modification tox as well. And we're also doing some work in that space. And there's been quite a lot of papers and interest in understanding what the proteins are that bind off target to these and what the consequences of those are. And actually, when we look through, it's not a random list of things, is that they're actually all RNA binding proteins. So RNA binding proteins are required to do everything to do with RNA and control gene expression and everything. And what happens is, you can see on here, you control your response to cold, to heat, to hypoxia, to DNA damage. They're all controlled by RNA binding proteins. And all these proteins have been found to bind to ASOs, which you really don't want, because you're stopping the cell's normal function. So what the ASOs are doing in some of the tox, it's like a saturation thing again, but you're taking away a normal cell function by the binding of the ASOs when they've gone in. And remember, when they're giving them to patients, it's 284 milligrams is going into the liver. This is really a very, very big dose that's going through. Okay, so we'll come back to the last bit. So this is in vitro transcribed RNA. In this case, it's the RNA is the prodrug. So we're not trying to knock out the RNA, we're trying to make more of a particular sort of protein in this situation. And you can use it for vaccination, and I've shown the example of the safety things that we've done on the COVID vaccine RNAs, but also you can use it for hormone replacement therapy. So instead of giving a protein, you can give an RNA, and the advantage of that is if you get it right, it can be more controllable, you can have a longer lasting dose effect. For things like rare diseases like leptin, which they would give a protein for that, that's very, very hard to make leptin, but actually it's very, very easy to make the RNAs. They're easy to make and very programmable, very, very cheap to do. And this is an exciting opportunity. So for those biologists there, so RNAs are read by these molecular machines called ribosomes, and it trundles along reading the genetic code 3 nucleotides at a time, and you end up with your protein product at the end. So that's how they kind of work. So it's the same problem again that we've had with the ASOs, in that you activate the innate immune response, and I'll just show you how that works. So it's very appealing in a therapy. You harness the cell machinery to make your drug. So the RNA is a prodrug. It's very cheap, it's rapid, and it's very modifiable. But again, the cell sees the RNA as foreign. But the Nobel Prize in Medicine was from Kathleen Carrico and Drew Reisin, who found out that if you modify the RNA with something, for them, it was actually a normal base within the RNA, pseudouridine, you put that in instead, that overcomes the impact of the innate immune response, which meant all of a sudden, having not been able to use RNA as a therapy, you can use it as a therapy, which was transformative, and that's why they won the Nobel Prize. And this is just showing some of the types of here. So I'll just stand in front of here and use my pointer if I can make this work. So this one, this is what's used, this is unmodified RNA. If you put that into a cell thinks it's being attacked by a virus, so it turns off its ability to translate the RNA, it stops it. That's part of the antiviral thing. If you modify, this is N1 methyl pseudouridine. This is not the one that was used in the trial. This is pseudouridine here, that works. This works even better, which is why BioNTech, Pfizer and Moderna are using this, and they're subject to the most enormous patent fights going on between them and Drew Weissman and Kathleen Carrico at the moment. So this works best in cells to make the most protein. Now this is a cell where you've just treated with RNA, and the reason it's going down is the cells die. So you've got a viral attack going on here, so the cells die. This is when you put it in, transfect the RNA into a cell where you've modified it with N1 methyl pseudouridine, the cell carries on growing. So it's very, very dramatic difference it makes by having that modification in there. So that's a good thing. So again, RNA alone, cell thinks it's being attacked by a virus. You modify the RNA, you take away that problem. And these were incredibly successful. So if you look at the efficacy on there versus a placebo, you know, you can see the red line going flat. So you really, really stop viral infection from this. And, you know, it has saved millions of lives. And if you look at the number of doses given by BioNTech, it's a huge number of doses. The most enormous thing has been done in the rollout of these medicines. So actually, because there have been some off-target effects in some individuals, but as a medicine as a whole, these are incredibly safe. If you look at the billions of people this has been given to, there's nothing else been rolled out quite in this way. But we were wondering about what the off-target things might be, not for the COVID vaccine, but if you want to use this in future medicines, and particularly repeat doses. So we were asking, this is different. It's not the same as a normal basis. So how does the ribosome, the reading machinery, sense having one N-methylpseudouridine in there? So what happens is, remember, the genetic code's triplet. If the ribosome comes along and finds a bit it's not that familiar with, it will stop for a while. And it'll stop because it hasn't got a bit to pair with it in the decoding. And when it stops, it kind of jiggles around a bit, and then it will shift along to make it to be able to read the next thing it can read. And that's called a frame shift. So you come along, you might pause, and you pause too long, it will stop and then go across. So normally, this would be, you're reading in a triplet, so AUG decodes methionine, that's the first base of the code there, and then you read along that way. If you shift by one, it's called a +1 frame shift, so you've gone along by one, so you don't read AUG, you read UGG instead. So you have a different amino acid put there instead. So you can have a correct protein made, or something that's complete garbage, because it's not anything you're expecting. So that's what a frame shift event does. Okay. So we set up this way of looking for frame shift events by looking for production of luciferase. It's a protein that gives you light as a reaction. So we could put these things in, and so we set it up that if you didn't do the frame shift, you'd make half a product that was inactive. If you did shift across, you could make something that had the end bit with 2 bits there, and that was functional. So that's how our assay worked. Don't worry about that. And what we found was when you have pseudouridine in there, you get lots more of this active light protein being made. So M1 methylpseudouridine induces +1 frame shifting. So you make what you think you make, and then this allows you to make something, but actually what happens in an ordinary situation. And this is what you can see on here. So you're making, the bottom band is the truncated bit, the top band is when you've made it and you frame shift it. So actually it's happening in quite a high frequency. It's not a rare event. It's about 8 to 10% we're having in there. We can put this through a machine and we can sequence all the protein in there, and we can work out at what point the ribosome's done its switch. So we say you're making normal stuff and you're making something random, and that's what the green is, what you would expect, and the red is random stuff. And what it allowed us to conclude is that for the scientists here, it's not pairing correctly with the methylpseudouridines, the tRNA that comes in. So the tRNA is a bit that brings the connection between the RNA and the protein. It's the in-between molecules. You've got an amino acid on the end, and you've got a decoding bit there that's marked in the fluorescent yellow. So that's the whole problem. You've got this sort of whole thing going on. When we looked through, it was matched our predictions. It's where we have runs of pseudouridine that we're getting frame shifting, and we're able to get a junction peptide between that had one bit we want and one bit we didn't, and they're fused together in the middle by the ribosome doing this shift across. Okay. So what about BioNTech vaccine? So what we did, we set up an assay where we made lots of peptides, bits in frame, and whole chunks of things out of frame, and we have a way by looking spots on a plate, it's called an ELISpot assay, are detecting, are we making the thing we think we're making, or are we making something else? So my colleague James did this experiment in mice. So he vaccinated mice with either Chadox, so that's the AstraZeneca vaccine that's not an RNA vaccine, it's a viral vector vaccine, or the BioNTech vaccine. And then you ask them, how good are you at recognizing the protein that's made in the spike protein from the coronavirus, from the virus, from the COVID, from SARS-CoV-2, the COVID virus. And you see that in both, they work pretty well. They're both pretty good vaccines in our animal model. That's really good. He then looked at where you get frame shifting. And actually, what we find, we get a very high rate of frame. So we're making, so the mouse immune system is actually, it's extraordinary that this works. So the RNA is being mistranslated. You're making something random. It's being processed and being presented to the immune system. So there's a T cell response within the immune system to the frame-shifted peptides on there, which was quite a surprise. What about people? So this was quite hard to get people in the end who'd only been vaccinated with the RNA vaccine versus the AstraZeneca vaccine, but we got 20 of each from our colleagues. So again, both vaccines work brilliantly against the SARS-CoV-2. But in individuals treated, we have a response to the frame-shifted peptides. So it means that frame shifting is happening in humans, and that there's individuals who've been vaccinated, they're making, well, this is a T cell response against the virus, but you're also making a T cell response against something random. And it was fine in this case. It was absolutely fine. We have no evidence of any harm. But if you'd made a T cell response to, say, a protein on your heart tissue, then you could actually cause autoimmunity by not correcting. Okay. So, but what you can do is you can change the code. You can recode these. We know where the frameshift sites are, and actually make them different. So, if you look on maybe the right-hand side, the best way of doing it, I showed you the frameshift you have this higher band. If you change the code, there's more than one code for UUU. You can put another code in there instead for leucine. And when you do that, you can get rid of it. If you get rid of both frameshift sites, you can remove it altogether. We can actually do the same on the COVID vaccine. So if you look on the top, that shows what you expect. This is just different chunks of the COVID vaccine. This is the bottom bit showing when you use 1-methylpseudouridine, you get the frameshift sites in there. If we recode it and take out runs of U's, but we still make the same, it still codes the same amino acid, we've just recoded it, our mutant version is safe. So you can recode these things to change it. So that's having really, really detailed tox understanding allows you to delve into the mechanism and make these newer things safe. But you need to be asking the right questions at the right time of how these things are working. And this was published in Nature about a year ago, and that was us celebrating. And this was my colleague James and Tom, who was a PhD student who did the work, and then two of my colleagues who worked closely with in Kent. But also we can use our knowledge to actually take this and put it in computational design, so we can have design algorithms for much safer things, and we can do the same for ASOs as well. You just combine it with a whole load of AI stuff, and you can actually train things to have things better. And so we've actually recoded the vaccine, and we put some other changes in as well, so that actually by having an understanding, we've got on here, so we've got our Pfizer vaccines in pink, and our optimal one, the one we've designed, is actually in red and green on this slide. So if you look at this, for the same dose, we've got a 20-fold better B cell response, so you make 20 times better, more antibodies. We've got about a 5-fold better T cell response. So it means you can bring the dose of these things down much more as well by really understanding the biology. Right, it's now 10:02, and I said I'd wrap up about then. So, summary. Programmable nucleic acid therapies have got potential to transform healthcare. I'm hugely excited by the opportunities that this will provide for everybody. But we've got to have a better understanding of the safety liabilities to allow their transition from treatment of rare to common diseases, but also for better treatment of rare diseases, to stop the failure rate. And the key concepts that I showed you at the beginning of dose, risk-benefit ratios, off-target tox, all still applies to these nucleic acid therapies. The same things apply. You've got to have the same understanding of them, but it's all about the mechanism. Got to drill down, understand the molecular cell biology, exactly what they're doing to the cell machinery, what they're binding to. And once you do that and really understand the mechanisms, the same way as the whole toxin I showed at the beginning, you can then start ameliorating the impact. So actually, tox in general, AstraZeneca have become very successful because they do their toxicology up front with drug development. And this is the same thing. So it's not one saying, oh, this is a disaster, you can't do this again. It's got to be an iterative process that you do it, you understand the tox liabilities, you redesign, you go back, try again. So it's a circular process, but bringing both things in at the same time, which is why tox research is really, really important. And so what we want is this safe by design agenda. So you make them safe up front. You have all this knowledge, you put it in your algorithms, you use your AI, you use your machine learning, and you design the very best thing that you can. It'll still need to be tested, but you'll be much closer to getting a product that's not going to fail at late stage or even early stage trials, which, for patients with rare diseases, it must be so devastating to have the hope of some sort of treatment to then have a trial failure. And we're dealing with some of that as well. So I couldn't have done the work without my really lovely lab. We're working on various aspects of things. So let me think. We've got Gavin and Veronica and Marguerite and Alex. They're all working on antisense oligonucleotides, various aspects of things. I've got Mark and Rob and Tom, and Tuya are working on the modified in vitro transcribed RNAs. We've got lots of things in that space, trying to make them safer, looking at design improvements. And then Eve, at the moment, are personally working on CRISPR-Cas9 editing systems on those. And then science should all be about collaboration, and that's when you do the most fun stuff. And I couldn't have done this work without James at the top left, and his postdoc Maria, or Mark and Tobias, which are below, with their postdocs Satish and Fabio. And of course, we're always really grateful for the funding that we have and makes our work possible. And thanks for listening, and happy to take any questions.